Right, back to the next stage of meiosis, which were known as the last stage of prophase 1. The prophase 1, the diakinesis. So the diakinesis. So at this stage, what happens at the diakinesis stage is that you still have your nucleus, you have your cell membrane rather, with your nucleus. This is the last stage, the late prophase. The nucleolus has started to disappear, and you have your chromosome. The chromosome try to repel each other, though having formed a cross-like structure. The chiasma is still visible, but the chromosome tries to repel each other. So you can see much more repellence in the chromosome. They try to repel in the sense that they move apart. They move apart. So you can see the two chromosomes trying to repel each other. And mind you, they've exchanged genetic material. They've exchanged genetic material. They've exchanged genetic material. One. So let me give it a good color here. So you can see, so this is a good pictorial. It's not, basically it's not actually very standard, but to some extent to give you information about what you're seeing in meiotic division. So you can see the exchange, you can see the blue chromosome and you can see the red chromosome. You can see the blue one is having red chromatids, and you can see that the red one also is having blue as a result of the crossing over that has occurred. This crossing over leads to genetic variation. We've talked about that. The crossing over leads to genetic variation, and it happens at the prophase 1. So the diakinesis stage, what characterizes diakinesis is that chromosomes move apart, moving apart of the homologs. So you have them moving apart of the them to repel each other, though they've exchanged genetic materials. So they are trying to repel each other, preparing for the next stage of division. So the chromosomes repel each other, preparing for the next stage of division. You can also see here, that there's an exchange. There's an exchange of genetic material here also. So there's an exchange of genetic material, but the chromosome tends to repel each other. They are moving towards, and the nucleolus has disappeared. The nucleolus has disappeared, and also we have the formation. We have the formation of spindle fibers. The spindle fibers tends to form. You have them forming. You have your spindle fiber. You can see them forming. So, and after the diakinesis stage, after the chromosomes has moved apart the homologs, like you see in the picture on the board, you can see that the chiasma also is still visible. You can see the chiasma is still visible, but what is unique about this is that the spindle fiber that starts forming is highly pronounced in the next stage of the meiotic division, known as the metaphase. From our interphase 1, your interphase does not have 1 or 2, but from your prophase 1, the chromosome enters what known as metaphase 1. And note, in any metaphase of any division, what characterizes it is that the nuclear component has disappeared, and there is free mixing, there is mixture in the components of the nuclear membrane. So what characterizes a metaphase stage, either in a mitotic division or a meiotic division, is just that you have the formation of spindle fibers. The spindle fibers has formed, the spindle fibers has formed and your nuclear membrane has dissolved. You have the formation of your spindle fibers and nuclear membrane has dissolved. Also, like I said, to avoid mise en place, rather, which means to put everything in order. So we need to put the next phase on the board. The next phase which is our metaphase 1. So metaphase 1, like we said, what characterizes any metaphase stage is spindle fibers formation. We have the formation of spindle fibers. And also take note that the nuclear membrane has dissolved. And what is happening is that the chromosomes lies at equatorial plates. So the chromosomes lie at the equatorial plates of the cell. From the word equi is meaning the central, the main, the middle of the cell. So that's what happens. You have the formation of spindle fibers and chromosome. The homologs, they lie at the equatorial plates. Don't forget that a chromosome has sister chromatids. A chromosome has 2 sister chromatids, and these chromosomes is joined together by what we know as a protein-like structure, centromere. So the centromere is what allows it, with the help of the kinetochores also, to move and migrate on the spindle-like fiber. You can just see, if you've seen some movements, you can see like the video I watched some years ago. You can see a man moving on a rope. You can move on a rope with the help of this structure that can just allow him to glide over the rope. He was able to glide over the rope because there is no friction, or because there is no restriction, so he was able to move on the rope. This also applies to the metaphase stage. The metaphase stage, you have the spindle fibers formation and chromosome lie at equatorial plate. So this is just a pictorial diagram. So this is your nuclear your cell membrane. I don't know why I get that in my mouth often. Sorry. And the next one we have is inside. So you should know that that's wrong. We have the formation of spindle fibers. The centrioles has moved towards the opposite poles, and you have your spindle fiber formation. So you have your spindle fiber formation here. So mind you, the chromosome has exchanged genetic structure, one having red and one having blue. So you can see that the chromosomes lies on the equatorial plate, ready to separate. So we are using a different and please take note, crossing over, I'm just explaining the crossing over on the first set of the chromosome that you have lying at the equatorial plate. They are the one that has crossed, they've exchanged genetic information. So this is a pictorial diagram of what happens at the metaphase. You can see your spindle fibers. You can see your spindle fibers looking like the rope. The spindle fibers. And you can see your chromosomes, each lying at what is known as the equatorial plate, the center of the cell. So that's an alignment in the central region, and you've seen all the centroles also are towards the opposite poles. When you see centroles towards opposite poles and with the formation of a rope-like structure called the spindle fibers, you know that you are the metaphase stage. So we see in the next video.